can start.
Okay, all right. Hi, everyone. I'm sorry for the complication and sorry for not being able to make to the conference in person. So today I will talk about scDisInFact, which is a disentangled learning method for integration and prediction of multi-batch and multi-condition single-cell RNA-seq data. So in this work, we are also considering data from multiple donors. We are considering only single-cell RNA-seq data from multiple donors. Single-cell RNA-seq data has been obtained to study disease in many different applications. So in these cases, we have donors which are from different biological conditions. So by biological conditions, I mean that patients can be in different disease severity levels, and they can have different drug treatments. There can also be other conditions like age and gender of the patients, etc. So from these donors, we obtain their samples, and then we obtain the single-cell RNA-seq count matrices. So once you get these count matrices, a question that you might consider is that should I integrate these matrices or not for the downstream analysis? The problem here is that between two count matrices, the difference can be a mixture of batch effect and the biological variation that is associated with the different biological conditions. So if we perform brute force integration, you might remove the interest information, so which is the variation caused by biological conditions. If you don't perform integration, then you might be concerned that there's batch effect. So we have designed scDisInFact, particularly thinking about this problem. So now in order to understand better what are the differences that exist between different count matrices, I have arranged these count matrices into this grid where rows correspond to different condition labels. So here I'm considering the disease severity condition where I have 3 labels: healthy, mild, and severe. And I have data from 6 different batches in this tau example. We know that between data from different batches, there exists batch effect. And I also call the variation that is caused by different biological condition labels, I also call that condition effect. Now, if I use some mathematical notations, which is not completely rigorous, to illustrate this idea, you can see that I have matrix A11, A12. These 2 matrices are in the same condition, but different batches. So the difference between these 2 matrices, I consider that is batch effect, which I also call batch factors. And this is the information that basically this is the noise that we want to remove. And if you look at these 2 matrices, A11, A21, the difference between them is biological variation because they are from the same batch and different conditions. So I call this difference the unshared. And also I call what the common information that is shared among all the matrices, I'll call that the shared biological factor. And that usually corresponds to the common cell type structure that is shared by all the data matrices. So now that I have defined these different factors, the batch factor, the unshared biological factor, and shared biological factor, scDisInFact is designed for 3 different aims, where the first aim is the most important one, which is to perform disentanglement of these 3 different types of factors. So we want to disentangle the shared biological factor, which is also called shared biofactor, and unshared biofactor and batch factors. Now, once the disentanglement is performed, that will enable us to perform 2 other tasks. One is key gene detection, where we detect the condition-associated key genes for each condition type. And the third task is perturbation prediction, where we can predict the gene expression data under unseen conditions. So task 2 and 3 are basically enabled by the success of task 1. Okay, so next I will show our model, excuse me. I will show our model that allows us to perform these 3 tasks. So this model is based on variational autoencoder model, where I have encoding networks on the left and decoding network on the right. So you can see that in the encoding networks, I have a shared encoder, which learns the shared Bao factors that is shared among other data matrices. And I also have an unshared encoder that learns the unshared Bao factors that particularly correspond to one condition type. Now, in my previous example, this condition type will be the disease. And then the decoder will take the shared Bao factor and unshared Bao factor and the batch factor, which is encoded basically the batch ID that is encoded in this batch factor, and it will reconstruct the original input data. So here I have the 3 factors, the shared Bao factor and the unshared Bao factor and the batch factors. So the 3 tasks, number 1 is disentangling. So once I learned these 3 types of factors, I can, that means I have performed disentanglement, and at the same time, I can remove the batch, also performs the batch effect removal. And the K-gene detection is performed in the unshared encoder that is particularly corresponding to a condition type. So for example, in this, from this unshared encoder that corresponds to disease severity, I can use feature selection procedure to get the genes that are associated with disease severity. And finally, perturbation prediction is performed in the decoder part by using different combinations of the learned shared Bao factor and unshared Bao factor and the batch factor. So that will give me the perturbation prediction results. Now in this figure, I'm showing only one unshared encoder. So that means I'm going to consider only one condition type, which is disease severity. Very often in our data, we have more than one condition types, right? So in this example, you can see condition type 1 is disease severity, and condition type 2 is drug treatment, for example, right? So I can have different condition values that is like treated and untreated. So in this case, I can generalize my previous model to handle more than one condition types by using multiple unshared encoders, where each unshared encoder corresponds to one condition type. Okay, so next I'm going to show some results from this model. I'm going to show results on 2 data sets. One is on a GBM, the glioblastoma data set. On this data set, I'm going to show results on task 1 and 2. And the second data set is a COVID-19 data set, where I'm going to show results on task 3. And in this COVID-19 data set, I have also 2 condition types to consider. So first data set is a glioblastoma data set, where I have got data from 6 different batches and 2, so 1 condition type that is drug treatment, and 2 condition labels, which is with drug treatment and without drug treatment. So before I apply scDisInFact, I just want to look at the original data to see whether there indeed is batch effect and a condition effect. So if I visualize the data with the batch, we can see that the batches are kind of separated, so they are not very well aligned, so which means there is batch effect. And if I visualize data with if I color the data with the conditions, we can also see that there is difference between different conditions. And that means both batch effect and condition effect exist in this data set. And next, I apply, after I apply scDisInFact, I'm going to visualize the shared Bao factors and unshared Bao factors to check visually whether my disentanglement has worked. So when I visualize the shared Bow factors, so our expectation on the shared Bow factor is basically similar to what we would like to see with the batch removal methods, right? So we want the cells to be very well separated by cell types, which is the case in this visualization. And we want the batches to be mixed, which is shown in the second UMAP plot. And next, I want to visualize the unshared Bow factors. Remember, the unshared Bow factors correspond to the difference that is caused by different biological conditions. So the unshared Bow factors should also be removed of batch effect. So that is what you see from the plot on the right, where we see that the batches are very well mixed. And then if I color the cells by different condition labels, we can see that the cells are separated by conditions. So this ensures that the disentanglement has worked and we have learned the factors correctly. And then we can go ahead and perform other tasks. So this result shows the results on how we can detect the key genes that are associated with the different conditions in this data set. So the key genes are obtained by a score that we have designed in our method. And the genes with top scores are more likely to be the key genes. So in this violin plot, I'm showing all the scores of the genes, and the genes that have high scores, which are highlighted by blue and red dots, are known markers that are associated with biological processes in this data set. And also we performed gene ontology analysis on top scored genes. And we observed that we can find the geo terms that are very relevant. So on this data set, I have shown results on disentanglement and paging detection. Next, I'm going to use a COVID-19 data set to show results on perturbation prediction. In this data set, we have collected data from studies, and we consider each study to be one batch. And then we have these conditions where we consider both disease severity, which has 3 levels: healthy, moderate, and severe. And we also consider age groups. So we have in total these many disease condition labels. So we want to test the ability of scDisInFact on predict data in unseen conditions. So to create unseen condition, I completely removed data from one condition combination, which is data from this column. So this data is used only in the evaluation process. And basically, I can use any other matrix to predict matrices in this unseen condition. So then we evaluate the prediction results by commonly used metrics like cluster-specific MSE, Pearson correlation, and R-squared scores, and then compared the scores with existing methods for perturbation prediction, and we observe that we have the highest score. This is mostly because that we have considered both batch effect and condition effect in our model. So to conclude, scDisInFact is a multitask tool to study disease and patient's response to drug treatment. And it can perform 3 tasks: disentanglement and batch removal, condition-associated gene detection, and perturbation prediction. I also want to point out that there are challenges in this kind of framework. Like if you give me data where batch and conditions correlate, for example, there is only one condition in one batch. That is basically some of the scenarios that the method would not work well. And so this basically implies that in experimental design, so this is not new, this is not proposed by us, it's already proposed in previous studies, that batches should be distributed across conditions. So finally, I want to acknowledge my team working on this project. So this work is led by my PhD student, Xiuwei Zhang, and collaborated with Dr. Peng Xiu's group at Georgia Tech with the PhD student, Xiuwei Zhang. And the project is supported by funding from NSF and NIH. I also want to advertise two other projects from our lab that is at ISMB. One is a poster presentation that is on, actually it's towards the end of the day, so I hope you got a chance to look at our poster. I see Multisim, which is a simulator of multi-omics single cell data and with ground truth cell-cell interactions and gene regulatory networks. Another project is a preceding presentation that is going to happen tomorrow. This is by undergrad student Mihir Bafna on prediction refinement of cell-cell interaction and gene regulatory networks. Okay, so thank you everyone for your time and attention, and I'm ready to take questions.
Thank you, Xiuwei. Is there questions from the audience?
Thanks for the cool talk. I have a question about the kind of like the assumption that you have. So you assume that the batch is invariant across multiple environments, and then you could isolate the other source of variation, which would be disease, cell type, and other things. I'm just wondering, like, isn't that a strong assumption? Because, and are those like other, let's say, I can't recall what was the name, like shared or unshared, are they actually identifiable? Because you actually look at the genes, and those genes could be correlated across multiple conditions. Because whenever you have like a healthy versus disease, like all the diseases are basically correlated with the batch.
Okay, so I'm not sure if I completely understood the question, so maybe I can use this illustration to kind of explain this. So the assumption is that so basically once we obtain like data set, so what we do is that we collect data from multiple studies, right? So and then for from these multiple data matrices, so batches are labeled by either different studies or by the original labels in the studies where they already have different batch labels. So yeah, so the assumption is that there is batch effect between different batches. And because we often see that data from different biological conditions are different, so we want to use the unshared biological factor to capture this difference. So and then from healthy to disease, you're right that if there are differences between genes, right, between gene expression levels, and hopefully that would be captured by this unshared biofactor. And then the shared biofactors would correspond to the common cell types that is still like existing in across different conditions. I'm not sure if I completely answered your question. I kind of missed a few words from your question.